Today, I'm going to talk about target repositioning, a novel method to predict therapeutic targets for various diseases. First, I will explain about the role of therapeutic targets in the treatment of diseases. Diseases are caused by dysfunction of the biological system consisting of genes, proteins, and pathways. The left figure shows that the biological system of a healthy subject is maintained by cooperation of many genes. The right figure shows that the gene network of patients is impaired. This impaired state can be recovered by regulating biomolecules related to disease causality and progress. These biomolecules are referred to as therapeutic targets. Therapeutic targets can lead to therapeutic effects for disease treatment. Identifying therapeutic targets is the first step of the drug discovery process. In this step, the associations between diseases and proteins are evaluated. The selection of inappropriate therapeutic targets reduces the success rate of clinical trials. However, therapeutic targets that can be identified using pathological knowledge have been thoroughly investigated. So, developing computational methods to predict therapeutic targets is needed. We propose the novel concept of target repositioning to predict new therapeutic targets, indications, or a wide range of diseases. For the prediction of therapeutic drugs, drug repositioning is popular. Drug repositioning identifies new indications for known drugs. The safety of known drugs has been already validated. So, drug repositioning is an efficient drug discovery approach. We propose the extension of the concept of drug repositioning to target repositioning. The bottom figure implies that target proteins have the potential therapeutic targetability for different diseases than the original diseases. Target repositioning predicts new indications of known therapeutic targets. The purpose of our study is to develop a computational method for target repositioning. We try to predict novel therapeutic targets for various diseases. We propose two methods by using disease-specific transcriptome data and target gene perturbed transcriptome data. First, we constructed disease-specific gene expression signatures. We collected gene expression profiles of patients with various diseases and averaged these gene expression profiles. We constructed a disease-specific gene expression signature for each of the 79 diseases, including Alzheimer's disease and breast cancer. Next, we constructed two types of gene perturbed transcriptome signatures for target proteins. Target gene perturbed transcriptome signatures represent gene expression patterns following gene knockdown and gene overexpression of target genes. We used gene knockdown signatures for 4,345 target proteins and gene overexpression signatures for 3,114 target proteins. We predicted inhibitory targets and activatory targets separately. Inhibitory targets can lead to therapeutic effects by inhibition, and activatory targets can lead to therapeutic effects by activation. For inhibitory target prediction, we used target gene knockdown signatures and disease-specific gene expression signatures. For activatory target predictions, we used target gene overexpression signatures and disease-specific gene expression signatures. Next, I will show our proposed methods. The first one is inverse signature method. Disease states are characterized by impaired expression patterns of genes. Inverting disease-specific expression patterns can lead to therapeutic effects. So, the optimal therapeutic targets are assumed to invert disease-specific gene expression patterns. We focus on the inverse correlations between target signatures and disease signatures to predict therapeutic targets. We predicted target proteins with highly inverse correlations as candidates for therapeutic targets. The next one is trans-disease method. We consider similarities among diseases to predict therapeutic targets. We assume that there is a known therapeutic target for disease A, and disease A and disease B is similar in terms of mechanisms. In this situation, the target for disease A can also have therapeutic effects for disease B as well. So, in the trans-disease method, we predicted therapeutic targets by multitask learning with disease similarities. In this method, we used target gene perturbed expression signatures as input data. Next, we prepared a predictive model for each disease. Each predictive model classifies input targets according to whether they are therapeutic targets for the disease or not. For trans-disease analysis, we learned predictive models jointly by sharing disease similarities. Finally, we predicted novel therapeutic targetability for diseases. I will explain the algorithm of our proposed method. We adopted a linear function that has weight vector W and explanatory variable X. We used target gene perturbed expression signatures as explanatory variables. We estimated weight vectors simultaneously by solving the following minimization problem. The first term is a logistic loss for all therapeutic targets. The second term is a ridge regularization, and the third term is a regularization term reflecting disease similarities. We designed the third term to bring the weight vectors close to each other if the diseases are similar. We evaluated the performance of our proposed methods. We compared proposed methods and baseline method. We adopted SNP profiling method based on SNP information as baseline method. In the SNP profiling method, we predicted susceptibility genes identified by GWAS analysis as therapeutic targets. The left figure shows the result of inhibitory target predictions, and the right figure shows the result of activatory target predictions. We evaluate the performance using the AUC, where 1 is perfect inference and 0.5 is random inference. Each box plot represents the distribution of AUC scores. We compare the performances between baseline method, inverse signature method, and trans-disease method. You can see that trans-disease method performs the best for both inhibitory and activatory target predictions. We performed target repositioning by using trans-disease method. Here is a network which represents the distribution of inhibitory targets repositioned from the original disease classes to other disease classes. Each node represents disease chapters in International Classification of Diseases. In the inhibitory target prediction, many inhibitory targets were repositioned between neoplasms and immune diseases, and between neoplasms and nervous system diseases. This result suggests that target repositioning can predict novel uses of existing inhibitory targets for different disease classes. we examined the distribution of activatory targets. Many activatory targets were repositioned between neoplasms and endocrine, nutritional, or metabolic diseases, and between endocrine, nutritional, or metabolic diseases and nervous system diseases. This result suggests that our proposed method has the potential to predict new therapeutic indications. This figure shows a small part of newly predicted inhibitory target disease association network. For example, TSLP is an inhibitory target for atopic dermatitis. TSLP was predicted to lead therapeutic effects in chronic myeloid leukemia, systemic lupus erythematosus, cervical cancer, ovarian cancer, and congenital muscular dystrophies. We performed literature search. We confirmed the validity of the therapeutic indications for 3 diseases with asterisks using independent resources such as recent papers and clinical reports. We also show the result of activatory target predictions. For example, we focus on newly predicted associations of ISMB. Using independent resources, we confirm the validity of predictions for 4 diseases: rheumatoid arthritis, breast cancer, type 1 diabetes, and acute myeloid leukemia. These results suggest that target repositioning can predict many promising therapeutic indications of existing activatory targets. So far, we predicted new indications for non-therapeutic targets. Finally, we tried to make predictions for orphan proteins without known target disease associations. For example, we focused on TAF1B. TAF1B was predicted to be an inhibitory target for adult T-cell leukemia. We performed geoanalysis by using TAF1B gene knockdown profile and examined the associations with adult T-cell leukemia. This network shows the result of geoanalysis. Circles and solid lines denote enriched geoterms and term-term relationships respectively. Geoterms of calcineurin and fat signaling cascade were enriched significantly. This signaling cascade is a causal pathway of adult T-cell leukemia. So, inhibiting TAF1B may lead to therapeutic effects for adult T-cell leukemia. Geoterms of leukocytes, including CD4 positive T cells, were also enriched. These cell lines are disease-associated cells. So, these results suggest that this prediction is biologically relevant. And these results imply that our proposed method can predict therapeutic indications not only for existing proteins, but also for orphan proteins without target disease associations. I will summarize our work. We propose target repositioning to predict therapeutic indications of existing therapeutic targets. We found that the proposed method is effective for both existing therapeutic targets and orphan proteins. Thank you for your kind attention.